welcome to our symposium on targeting and readout and non-invasive brain stimulation. My name is Thomas Knöscher from the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig, Germany. And I have the pleasure to hold this symposium together with my co-organizer, Axel Hirscher from the University of Copenhagen, as well as Daria Antonenko from the University of Greifswald and Gesa Hartwigsen, also from the MPI in Leipzig, Germany. So what about non-invasive brain stimulation? Of course, our goal is to understand the brain, in particular, how the brain is actually functioning. So how all the very complex components of the brain interact with each other to generate the brain's performances. And like with any other system that cannot be directly investigated, we use classically black box methods in experimental psychology, classical brain science. And this is done by using the channels, the stimulation channels provided by nature in order to stimulate the system, and then using the natural output channels like behavior, motor behavior, autonomous reactions like blood pressure or heart rate, as well as language, in order to observe the system's response. However, because the system is very complex in nature, these input-output channels might not be enough in order to understand and completely illuminate the mechanisms inside the brain. And for that purpose, it is advantageous to be able to look at the brain at work. So different positions in the brain, the activity at different locations in the brain. And this is where non-invasive brain imaging comes into play, which uses different physical principles to observe the brain at work. For example, EEG, MEG, fMRI, and so on. Likewise, also the stimulation of arbitrary parts within the brain outside the predefined input channels would be an advantage. And this is done by non-invasive brain stimulation, which is mediated by electric and magnetic fields that can be injected into the head and through the skull into the brain. Importantly, in order to take full advantage of these techniques, we have to understand how and where exactly the electric and magnetic fields influence the working of the brain. So where is actually the target, and how, in the end, these electric and magnetic fields influence the performance of the brain and the eventual observable variables, which could be non-invasive brain stimulation or behavior or anything else. So the readout variables. And in order to do this, we have prepared, or in order to illuminate this, we have prepared 4 different presentations. The first one held by Daria Antonenko. She will deal with electric brain stimulation. Electric brain stimulation is performed by attaching electrodes to the head surface and injecting a relatively weak current through the brain, which influences the excitabilities of cells and thereby brain function. Daria will first illuminate how electrode placement and the injected current on the one side and the geometry and anatomy of the head on the other side gives rise to particular distributions of electric fields within the brain, and then how these electric fields have an influence on the empirical measurable response. In the second presentation, I myself will present techniques that allow for the localization of targets of transcranial magnetic stimulation. Transcranial magnetic stimulation takes advantage of magnetic fields that induce electric fields within the brain. And as we can see here in this cartoon, these electric fields are not exactly focused. So with each stimulation, we have many different places that are stimulated. We want to know where exactly do we influence the mechanics of the brain. And this we do by observing some readout variable, like here, for example, a motor evoked potential amplitude, and then looking for different positions in the brain at the correlation between electric field and readout magnitude. And this helps us to localize the effects in the brain. In the next presentation, Axel Tischer will introduce a very interesting and novel technique that allows to validate the computed, the predicted electric field distributions from transcranial electric or magnetic stimulation using MR techniques. He will present MRI techniques that are capable of measuring on the fly electric fields in the brain. And finally, Gesa Hartwigsen will look a little bit at the network or systems level. So how does the immensely complex network of neurons and neuronal populations in the brain react to non-invasive brain stimulation? In particular, how simultaneous and prior stimulation, so stimulation before the readout, will influence networks in terms of adaptive plasticity and also short-term reorganization with the simultaneous stimulation. And in particular, she will deal with the reorganization of the language network. And with that introduction, I would like to hand over the stage to the first speaker, which is Daria Antonenko. Thank you very much.
In my talk, I will be speaking about individual differences in electric fields induced by non-invasive brain stimulation in the human brain, specifically transcranial electrical stimulation and its relationship to cognitive effects of stimulation. First, I will give a very brief introduction on what transcranial electrical stimulation is and how we can simulate electric fields on an individual basis using computational modeling analysis in the human brain of each participant. Second, these simulations can be used to extract individual field parameters that are impacted by anatomical features. And I will present a study where we try to disentangle contributors from head and brain anatomy. Third, I'll present an example of a link between E fields and the magnitude of neurophysiological and behavioral effects of transcranial electrical stimulation. And finally, I will give an outlook on how we can extend simulations from retrospective analysis to prospective planning of simulation experiments. Non-invasive brain stimulation has different forms. And the one mostly used is transcranial electrical stimulation. And here we use a different amount of electrodes connected to the stimulation device attached to the head of the participant to induce a mild current on the scalp that is thought to affect brain activity of the underlying cortex, either by affecting stabilization of neurons or, for instance, interact directly with brain oscillations. There are different electrode configurations that can be divided mainly in conventional and so-called high density or high definition setups. And while conventional setups are thought to affect large-scale brain networks, the newer high-density setups applying the current on more than 2 electrodes are suggested to have more localized effects on specific neural network hubs. Normally, currents of 1 or 2 milliamp are applied for approximately 20 minutes during or before task execution. Parameters such as polarity, placement, and number of electrodes depend on the research questions, thus which domain and neural network and regions are targeted. Computational modeling analysis can then simulate the current that is induced on the cortex using individual magnetic resonance images of each participant. These are normally structural 3D, T1 and T2 weighted images. Synapse here is a software tool that allows a very convenient conduction of these modeling analysis. And here each head is segmented into its compartments, namely skin, skull, cerebrospinal fluid, gray and white matter, to create realistic head models that can then be used to model the electric fields induced by a particular electrode configuration, thus the placement of electrodes of particular sizes. Here you can see how this can be realized when electrodes are attached to the head during an MRI experiment. Thus in the scanner. Of course, this is also possible if brain stimulation is applied outside of the scanner with independent acquisition of structural images. Importantly, these analyses show that there is variability between participants, meaning that the same application of current induces differences depending on the individual. Here you can see brains of different participants where we simulated the same configuration, which was a prefrontal stimulation in this case, while distribution is quite similar. Parameters such as maximum field strength and focality of the field vary between participants. This variation is most likely ascribed to head and brain anatomy of the subjects, but it is not resolved yet which tissue types contribute to it and to which extent also. In order to understand this, we modeled 6 different electrode configurations. As the conventional electrode setup, usually applied in clinical and cognitive neuroscience experiments. We extracted the general E-field strength, which we defined as 75th percentile of field magnitude. E-field strength was higher in young compared to older adults and almost all or in all configurations. Importantly, this was also true for average E-field strengths and also E-field strengths in a region of interest, but also for focality of the stimulation. This means that older adults showed more focal stimulation compared to young adults, with less area being stimulated with maximum current. Here you can see the mean distribution and standard deviation for all the configurations we selected due to the application and empirical studies of transcranial electrical stimulation. What we observed, most importantly, again, is variability on older adults and in young adults. So the amount of current that reaches the individual cortex of a participant varies between individuals and also between age groups. In our samples, for example, you can see it here, the variability was higher in young adults for all configurations, or here, the standard deviation. This may be different in other participant cohorts also. Field strength was highly correlated between electrode configurations, suggesting that similar factors actually may influence the induced electric field on the level of the cortex for different configurations. So you can see here all the configurations correlated in both age groups, very high. Right, so this suggests that similar factors may influence induced electric field on the level of the cortex for different configurations, and probably just similar mechanisms underlie the effects of transcranial electrical stimulations, irrespective of electrode location. So one conclusion of this observation could be that a participant showing plasticity with one form of TES application should also show plasticity induction using other parameters of stimulation. Interestingly, focality of stimulation, so the size of the area with maximum current field induction, did mostly not correlate between electrode configurations. This could mean that different factors determine the focality of given parameters of the stimulation. We were further interested in the impact of head and brain volume on E-field strength and found that in particular total head volume, and especially skull and skin and CSF volumes contributed to E-field strength. In addition, there was also an interaction with the age group, meaning that the relationship between E-field strength and head volume and CSF volume was weaker and older compared to young adults, which you can see here, the relationship in older adults and the relationship in young adults, which showed a steeper correlation. Right, so we can extract the E-field, general E-field strengths, E-field within region of interest, and focality values as parameters from the E-field simulation that vary, again, between individuals and also groups. The general E-field strengths can be defined as 75th percentile or also 19th percentile of maximum field values, and we can average E-field within regions of interest, given that we know actually we can define the target of stimulation. Importantly, all correlations with anatomical factors were similar regardless of which exact parameter we used. Though it might be useful to take global volumes of the brain, but also the head into account. Some studies, for instance, provide results for non-significant group differences in voxel-based morphometry as proof of no differences in anatomy, but still head size differences could affect the magnitude of response. Also, I think it is important to visualize the distribution of groups of subjects, for instance, in different stimulation intervention groups, and to visualize distributions for different patient cohorts. Next, as many studies actually seem to acquire structural T1 and T2 weighted images of each participant, those should be used to approach the question of a link to empirical effects. So, in addition to anatomical determinants of E-fields, it is of particular interest whether they can be predictive for any empirical effect of brain stimulation, thus magnitude of response, which can be on the neurophysiological level or on the behavioral level, for instance. Sorry. In our first study, where we used modeling, we first aimed to replicate previously shown effects on GABA levels and on functional connectivity of the sensory motor network in healthy young individuals. This replication was successful. We observed reduced levels of GABA within the hand knob following a nodal and cathodal transcranial direct current stimulation of the left motor cortex. Functional connectivity with the motor networks was increased in region below the two electrodes, you can see here. Importantly, when looking at electric field strength and also normal component of the electric field, we observed positive correlations in the following direction. The higher the electric field in a network hub that was actually affected by the stimulation, the more GABA concentration was modulated, and in this case reduced by transcranial direct current stimulation. And same for the functional connectivity. So the higher the electric field strength in a network hub affected by the stimulation, the more or the higher the sensory network strength modulation. So in this case, increase by a nodal transcranial direct current stimulation. In another study where we target specific networks with combined interventions of cognitive training and brain stimulation, we use modeling to link the induced field magnitude to behavioral responses. Here, in a rather recent study, we found that the individual benefit, thus the magnitude of response, to a 3-week cognitive training plus prefrontal electrical stimulation was positively related to the individual E-field strengths that was induced by the stimulation. And there was no correlation in the sham group. Thus, the more current was induced in the group where actual brain stimulation was applied compared to the placebo stimulation group, the more performance levels improved after the intervention. We use actually a transfer task, so not the training task, to measure these behavioral effects of stimulation. And you can see here that there was a minor group effect also in this task. So the nodal group improved more relative to the placebo group. And as I said, the magnitude of improvement correlated to the E-field strength. So finally, while current research is trying to understand variability in brain stimulation effects using computational modeling, these approaches may also be useful to plan simulation configurations before the start of an experiment. For example, what we recently showed is that head circumference, a potential proxy of head volume, was related to modeled E fields. Thus, the larger the head of a participant, the higher the head circumference, obviously, and the lower the induced E field on the individual cortex. The height of this relationship was quite independent of the placement of electrodes, what you can see here. And this relationship between head circumference and E field was also observed for focal montages. So electrode configurations where we try to target a more localized network hub, but it was weaker compared to conventional electrode configurations. This suggests that other factors such as regional volumes, probably, or skin thickness underneath the electrodes, already shown in some previous studies, may be more relevant in this case. This relationship may be particularly relevant in studies where there's no possibility to acquire magnetic resonance images of each participant, for instance. So far, there's also no adjustment of stimulation intensities in brain stimulation studies. Thus, mostly all follow the one-size-fits-all approach. And these simulations can now open up a possibility for both retrospective analysis of already conducted experiments, but also for prospective planning, for instance, individual intensities. Finally, yet importantly, I want to raise an important point when it comes to the aim of the comprehensive understanding of transcranial electrical stimulation effects. Recently, application of transcranial electrical stimulation using large electrodes with sizes of up to 35 square centimeters have been criticized because of the observation made also by computational modeling analysis that the electric field is distributed over large parts of the cortex, sometimes reaching several brain regions such as occipital, temporal, parietal lobes, with maximum current also induced between electrodes rather than underneath the electrodes. This has also promoted the conclusion that effects of conventional electrode configurations with large sponge electrodes may be unspecific or more unspecific than more localized electrode configurations. Although this might also be true, in a recent study we provided evidence that the effects may not be inferred on the level of the underlying anatomy only, but interact with ongoing brain activity during stimulation. So here we found that the application of a nodal stimulation over the right temporoparietal cortex for 3 consecutive days during performance of object location memory tasks led to very focal effects on the level of functional connectivity. Here we applied eigenvector centrality mapping analysis of resting state fMRI images acquired before and after the intervention and observed decreased centrality of temporooccipital brain areas after the combination of a nodal TDCS with object location memory training. This points towards targeted effects of brain stimulation when coupled with task-related brain activity and underscores that multimodal brain imaging is required for comprehensive understanding of brain stimulation effects. With that, I want to thank all my colleagues and collaborators and you for your attention. Looking forward to your questions either now or anytime via email.
So if you want to design a TMS experiment or interpret its results, we have to consider a chain of dependencies which start at the position, orientation, and geometric design of the TMS coil, which together with the geometry of the head determines the spatial distribution of the electric field over the cortex or over the brain, which in turn, or this electric field in turn, has a profound influence on the functioning of the neurons, on their membrane potentials, firing rate, excitabilities, and so forth, which naturally influence the neural dynamics, the behavior of neural circuits, which are causal for observable behavioral, physiological, or brain activity-wise variables. For example, the amplitude of a motor evoked potential. In this context, we are interested in a number of questions. The first one being target localization. We want to know where are the neurons, the stimulation of which is related to the observed or expected effects. This question is not trivial because with each stimulation, as we can see here, for example, in this picture, we reach a large variety of locations on the cortical surface with more or less intensity. Once we know the location, we would like to know how to dose our stimulation. That means which strength or field direction do we have to apply or achieve at the target location in order to get the effect we desire. And once we know all this, of course, we want to know how to achieve this. So which are the stimulation parameters, the coil positions and coil orientations, the stimulation intensities that we have to apply in order to get maximum or get just the right stimulation at the target location and minimum stimulation elsewhere. Here in this presentation, I would like to focus on target localization. And the most simple or classical way to get target localization is obviously exhaustive mapping. So on some grid, regular or irregular grid, over some region of interest, I test out all coil positions defined either as projections of the coil center onto the cortex or as locations of the field maxima, if I'm able to compute the electric field on the fly. And at each of these coil positions, I try out all possible coil orientations, of course with a certain spacing. And for each of these position orientation combination, I have to find the activation threshold by trying out different intensities. And this location and orientation, where I have the lowest activation threshold, is the one where the target neurons are actually located. So this procedure comes down to an exhaustive optimization procedure in 4 or higher dimensional space, which is either costly or imprecise or both. So the question arises, can we do better with fewer measurements or using or achieving higher precision? And an approach to achieve this is the so-called correlative target localization approach, which is based on the key assumption that at the cortical location underlying the induced effect, we have a unique relationship between the effect size, for example, the amplitude of an evoked motor evoked potential in the hand muscles, and the induced electric field at this location. Yeah, no matter which coil position and coil orientation I have used to achieve this electric field at this position. This relationship is mostly monotonous. So with increasing E field, I also get increasing readout variables. And usually it has this sigmoidal shape because the E field or the observable effect, the motor evoked potential, for example, cannot go below 0, but it cannot go beyond a certain maximal value either. So in order to test out this approach, we might use a very simple experiment. We stimulate over the motor cortex, exciting pyramidal cells to a more or less extent. These pyramidal cells in turn excite secondary motor neurons in the spinal cord, which project to the hand muscles where we can measure motor evoked potentials. We consider voxels in a region of interest that covers a wider region of the motor system in the contralateral hemisphere. And we locate the coil at different positions indicated by these balls and orient them into different directions indicated by these arrows. And our readout variable, as mentioned, is the MEP amplitude. So if you do that, then for each different combination of orientation and position, these combinations of orientation and position, we usually term a condition. So we apply different conditions. For each of these conditions, if I crank up the stimulator intensity, I get increasing MEP amplitudes. So this input-output curve I get each time, but it is shifted horizontally because some of these conditions, orientation position combinations, produce higher electric fields in the right location, in the location where the relevant neurons are located, and others not, which is due to the different field distributions caused by the different conditions, as we can see here. Yeah. So if I now can translate for each voxel, yeah, in each condition for each voxel, this intensity to MEP amplitude curve into an E field to MEP amplitude curve just by forward simulation. And the theory is now predicting that this E field to MEP curve in the right location should, all these curves should be more or less on top of each other. Because the same E field, no matter which way it is created, produces the same MEP. While at a location which is not responsible for the evocation of the MEP, these curves can be shifted. Yeah? So I get, depending upon which combination of orientation and position I use, I get different MEP amplitudes for the same E field. So this congruence of the different input-output curves can be measured, for example, by the amount to which I have to shift them on top of each other. And this congruence factor can be mapped onto the cortex and gives us the target location. If we apply this technique, first very modestly, using just 2 different positions and 3 different orientations for each position, we get the following results. Excuse me. In many subjects, we see here a magnification of the cortical surface of each subject. This is the central sulcus always. This is the precentral gyrus and the postcentral gyrus. And in a number of subjects, here in 8 subjects, we get what we expect. A peak, a hotspot on the precentral, on the crown of the precentral gyrus, just in the hand knob area. We can see that in each case, more or less. But there are also cases where we have two hotspots on the pre- and post-central gyrus, so in the motor and somatosensory area. And in some subjects, this is even more prominent in the somatosensory area. And we also had one subject where the interpretation was really difficult. So is this real? So is this reflecting some physiological reality? Or is it simply due to the low number of position orientation combinations, so that these 2 hotspots happen to be similar in terms of electric field for all these orientation position combinations, for all these conditions? Yeah. So in the light of these measurements, then these 2 hotspots wouldn't be distinguishable. So that question remains, and it can be investigated by just using 4 or more position orientation combinations. Yeah, so far we used 2 with 3 orientations in each case, and we got these results. I just picked of each of these 3 groups 1 example. Yeah, these are 6 conditions. If I now use 20 conditions with 4 different positions and 5 orientations in each case, out of the sudden I get something that is much more plausible. Yeah, I get a single hotspot on the precentral gyrus in the hand knob area in each case. Yeah, and that is really reproducible. So, however, this is quite costly. For the 6 conditions, I already need 1,000 pulses to map out these 6 I/O curves in each case. For the 20 conditions, even if I use fewer stimulations for each I/O curve, I still need something like 3,500 pulses. This is a lot. So the question remains whether I really need these 20 conditions, these 20 different position orientation combinations, or whether I only need the right ones. So would an optimal subset of these 20 suffice? And we investigate that by just drawing random subsets of different sizes from these 20 conditions. These are very many different combinations. Altogether, there are more than 1 million different combinations you can draw from 20 conditions. And that means that for each number of conditions, I have a whole range of performances. The performance is measured by the shape of the congruence map. If the congruence map is very peaky, then it's very selective. It really distinguishes between really nearby cortical sites. If it's more smooth or flat, then it's not so selective. And therefore, we just thresholded the congruence maps and measured the thresholded area as a measure of peakiness. And you can see that for each number of conditions, there's a whole range of quality measures. Yeah. If I take the worst case for each number of conditions, then this whole thing would improve all the way up to the 20 conditions. Yeah. So the worst case always becomes better and better and better if I use more conditions. While if I take the optimal case, the best combination of 6, 7, or 9 conditions, then already with 5 or 6 different conditions, I have optimal performance, even better than with 20. Yeah, this slight increase here is probably due to the addition of noise by these additional measurements. Yeah, so the question is then, how can we find these optimal conditions? And that is obviously an optimization procedure or an optimization task. And we need some correlate that we can use beforehand that would predict us the peakiness or the area of the resulting congruence map. And one such thing that one would suspect is the variability of the different field distributions of the field topographies. Because if the field topographies are very different between the different conditions, then they are probably capable of distinguishing between different cortical sites. So we investigated that, and we indeed got a certain correlation, yeah, which is clear, but it's not very unique. Yeah, so at a particular field variability, I can really get very different congruence maps. So this question of what a good criterion is for condition selection is still under research, under investigation. So let's look at the results. Previously, we used these 20 conditions, yeah, and we got these nice results. If we pick out the 6 best out of these 20 conditions, yeah, the most optimal combination, then we get a very similar result. So this confirms that it is not the sheer number of conditions, but the proper selection of them. So this is, of course, difficult to achieve because we have this problem of the right criterion. So there might be another way out. How can we improve the number of different field topographies that we apply without increasing the number of administered pulses exceedingly? So far, we use some sort of a 2-stage regression approach. Yeah, first, for each combination of position and orientation, we measure a lot of points, yeah, in order to get a reliable I/O curve by regression of a certain function. And then we measure the similarity between these I/O curves, yeah, for a certain voxel, yeah, which is a second regression, if you like. Yeah, so I look how much I can describe these different curves by just one curve. So if I now would, instead of measuring the I/O curves with many pulses for a few combinations of positions and orientations, I would just administer one pulse for each combination of position and orientation, yeah, and do that very many times. Then I get much more variability. Using the same number of regression points, I get much more field variability. Still, in each voxel, I would get different field magnitudes just by virtue of the different orientation position combinations, which produce different field maxima, even with the same intensity at certain positions. So we tried out that idea. One advantage being that now, because I use so many conditions, I can afford to choose them randomly, yeah, because I try to sample all possible combinations of positions and orientations. So we did so here in a region of interest, which has been defined circular here. And then we do only one regression for each voxel, yeah, the electric fields produced by all these different stimulations versus the MEPs observed for them, and do one single regression. Here we tried with different functions, linear, sigmoidal, or logarithmic sigmoidal, but I can tell you it doesn't make a lot of difference. It doesn't matter, as long as it is a monotonous function. Okay, and we get very convincing results, similar to the one before. For example, here you can see these hotspots. We even distinguished between 3 different readout variables, different MEPs for different finger muscles. And we find closely spaced but distinguishable hotspots on the precentral gyrus for these different fingers. We did so in many subjects, and we found some variability in these locations, their relative position to each other, but also a little bit of systematicity. Overall, the question remains a little bit what the real resolution actually is. This is something that still has to be investigated systematically. But what is even more interesting and important is, does that really save us time? And for that, we have to look. Now we administered 1,000 pulses, but we have to look how many pulses are actually necessary. So we did the same as we did before. We just drew samples of 10, 20, 100, 500 conditions out of these thousands many times, and looked how much the congruence maps deviated from the full congruence map with the 1,000. And we find a deviation of 5% as a good approximation, and found that to achieve these 5%, we need less than 150 random stimulations, opposed to 1,000 in the other approach. Because we don't know the full congruence map in a real experiment, because we don't want to deliver these 1,000 pulses, we want to stop at 100 or 150. We use the increment from one step to the other for that. And it turns out that these 5% here translate into a 2% change if I just add one pulse. So just wrapping up, it turns out the stimulation with random positions and orientations increases the information gain of each pulse, because it adds to the variability of the field patterns, leading to a much lower number of necessary pulses. And it's also much easier to perform, because we do not have to place the coil in defined positions and so on. However, it also turned out in simulations that optimal positions and orientations would indeed improve the thing even further. But that leaves the question how we could find these positions and orientations. So how to optimize beforehand the stimulation scheme and how to realize it. Realization means that we have to precisely adopt a certain position orientation combination. And that can be achieved by the aid of robotic TMS. And finally, we want to generalize to other experiments and readout types, other readout variables, and also to situations where multiple locations might influence the readout variables, which turns the univariate regression problem into a multivariate regression problem, which is, of course, a challenge. And with that, I would like to thank my collaborators, Konstantin and Ole, who did most of the work, but also Axel, Gesa, and Anna, who contributed, and you as an audience. Thank you very much, and bye-bye.
Hi, I'm Axel Thielcher. I'm working at the Danish Research Center for Magnetic Resonance and also at the Technical University of Denmark. And I'll talk about the online combination of non-invasive brain stimulation with MRI today. I will specifically cover two stimulation techniques, namely magnetic stimulation, transcranial magnetic stimulation, which uses electromagnetic induction to create an electric field in the brain. And this electric field then depolarizes the membranes of neurons, leading to action potentials. And that's most easily seen when you stimulate the motor cortex, because then you get clear muscle twitches, which you can record, for example, using EMG surface electrodes. The second technique I'll talk about is electric stimulation, which actually induces far weaker electric fields in the brain, which do not depolarize the membranes themselves, but just slightly modulate it. And then by that also modulate cortical activity. This has been shown already quite early on in a study in rats, where compared to spontaneous baseline firing of a cell, the spiking activity was either decreased or increased depending on the direction of the induced current flow relative to the cortex. the chain of events when you apply brain stimulation roughly looks like that. So of the immediate effects is that you have your electric field induced, which differs in strength and in pattern, which then leads to changes of the membrane polarization of the affected nerves or neural structures, which in turn leads to a local modulation of neural activity in the more strongly stimulated regions, changing neural activity there. And then again, obviously the output of this region changes so that you also then see network responses to the stimulation. And this is not just a one-way street, but these different levels interact with each other. So how does then brain MRI come into play here? Well, what we have is that we induce some electric field, and that's something we can try to calculate. But what we then are really interested to see is how this electric field then changes the physiological or physiology of the brain, or how the physiological response looks like. So, and there obviously functional fMRI gives a good readout of the effects on brain activity. More recent application is that you can also use MRI actually to measure the current flow, which is induced by electric stimulation. And this is extremely useful because so far we relied only on calculations to get some estimates on how the field would look like. But now we can also use MRI increasingly well as a technique to validate these simulations and make sure that they also fit. I will not talk too much about the technical and methodological details of these two techniques. I just want to point towards two recent papers. One is out as preprint, one is just published basically. This here is about TESS fMRI combination, which is a consensus study involving a lot of people. And this is a recent review we did on TMS fMRI. And I think both are actually great resources to get you started more on the technical parts of these techniques. The next few minutes, I want to talk about what you can actually use TMS fMRI for exactly, and give basically just two examples to demonstrate that it can be really useful. So first of all, we can use it to assess effective connectivity, simply because we can use TMS to induce activity locally in the brain region, and we can use fMRI then to see basically where it travels from to. And the interesting part is really that this pattern of functional or effective connectivity is not fixed, but it depends on the state of the brain. So for example, when we are sleeping, there will be a very different connectivity pattern compared to when we are awake, but also the connectivity pattern will change depending on whether we do one task or another task. And mapping this connectivity pattern is really an interesting challenge where TMS fMRI can help. That's actually was one of our first applications back in 2012. This was a study done by Marius Misa, at that time in my lab, now it's in Zurich. With the setup at that time, you see the TMS call. Here we have the lower part of the fMRI or the MRI birdcage call, which would then be around it. And what we targeted there at that study was the left premotor cortex, which is known to be an important node in motor control, especially to map external cues onto the appropriate responses. This was well demonstrated in animal studies. In fMRI studies in humans, the situation was a bit more unclear because it demonstrated actually these studies demonstrated relatively broad activity of the left PMD in some studies which did involve these kinds of mappings between cues and motor responses, but also in studies which studied more internally driven movements. So we wondered whether actually, even though the left PMD showed a similar level of overall bold activity, maybe its task-specific connectivity pattern differed, and maybe that's something we could actually test with TMS fMRI. So what we did is we contrasted two tasks against each other. One was associative task where they had the participants had to learn the mapping between a cue and the response. The cues were abstract, visual figures, and the people then had to respond with a certain finger press. The alternative was that we had a condition where people could freely select which finger they would press, and there the cue, were cues of similar pattern, a visual pattern, they just specified the response timing. We had two TMS intensities, one where we thought that it would be neurally effective, and a lower intensity to control for side effects like the auditory noise caused by TMS stimulation. As a stimulation pattern, we used always short bursts of 5 pulses, and this was repeated every 4 seconds, as demonstrated or indicated here over the task period, and then we had a rest period to follow. So this will become interesting later again. For now, let's focus on the behavioral effects. There were actually no effects of stimulation. There was a clear effect on the motor task, and that was actually expected, because in the task where they had to first see which cue came up to then select the proper finger, that just took longer as the case where they could freely select it, because the selection could be done already before the visual cue came up, and then once the cue came up, they just had to respond. So this was highly significant, but importantly, there was really no TMS effect, neither a main effect nor some kind of interaction effect. So behaviorally, TMS did not change the outcome, which also means that actually our fMRI readout of the TMS effects would not be confounded by behavioral changes induced between the different conditions by TMS. So in terms of main effects, we saw very stable activations in parietal frontal regions, all involved in visual motor processing. The interesting thing is that these stable effects occurred for both of the tasks, and there was really no significant difference of this activity between the two tasks. So this was the stimulated area with stimulated by TMS, so that's PMD, and this common pattern occurred stably for both tasks. The interesting thing was then to see whether actually the TMS effect on brain activity depended on which task people were doing at the time. So we looked for an interaction between both, and there was a clear interaction specifically in the right regions, motor regions like PMD, but also more a bit more central areas like SMA. And when looking into the contrast, then it became clear that actually the left PMD, which is around here, had the stimulation there had a clear-cut effect when people had to do selection tasks, basically selecting the proper response according to the visual cue. Then TMS had a very clear effect, versus when they could freely select, there was really no effect of TMS. So basically, first of all, we could demonstrate then that the mood effects of TMS were obviously task specific. The interesting thing is that this occurred even though the general activation levels in the studied motor network were pretty similar. And it was also then becoming clear that the left PMD had a stronger functional coupling to other areas of the motor network when it was involved in establishing the specific stimulus response mapping. So that, I think, shows quite nicely what the combination of both techniques can be used for. In a little bit later study, which is still in preparation, and which was mainly driven by my colleague Christopher Maassen, we followed up on that in terms of that we again studied the left PMD as our stimulation target, this time just in rest, and applied two pulse patterns. I will only look into one today now, which we studied at varying intensities. And a very interesting finding there was actually that in one of the two pulse patterns, which was a block design, meaning that we again, similar to what we did in the prior study, gave continuous stimulation through the complete block actually, but this time with slightly different or with different protocol TMS parameters, namely a stimulation with 0.7 Hertz. So we always gave 2 pulses here, as a brief interval, and then waited for around 1.3 seconds to give the next, and so on. That actually resulted, so this is the main effect shown here of across all intensities, that resulted in a very, very stable or decrease of activity this time. So meaning that, first of all, it means that when you want to use TMS fMRIs to study conductivity, then you have to carefully choose your TMS protocol, because you want to really more induce transient short-term excitatory effect on this stimulated area, which then can travel further. But on the other hand, it also shows that this method is extremely powerful to reveal more modulatory effects of the TMS on the brain state. And this is interesting because 0.7 Hertz is really quite close to 1 Hertz repetitive TMS, which is known as a inhibitory TMS protocol. So basically, you can also use fMRI here to reveal what TMS does to the brain on a short-term level within 20 seconds. That's quite exciting. Now, coming to, again, back to the slide here, I gave a few examples on how fMRI could be used to learn more about TMS, but also how TMS could be used to study effective conductivity. But actually, MRI can play a very different role in the combination with brain stimulation, namely as a validation tool to just validate whether our predictions, our calculations about how the electric field pattern looks like are true or not true. And for that, as there are new upcoming techniques called RCDI and MRCDIT, which stands for magnetic resonance current density imaging, or magnetic resonance electrical impedance tomography. So in principle, these techniques are relatively simple. What you do is you induce currents flowing through the brain by some external source. And these currents, when they flow through the brain, will also change the magnetic field there slightly. So you will change the magnetic field of the scanner in which the subject is positioned, and that's something you can actually measure. So usually you just feed the currents through in 2 directions, one time in one direction, then in the reverse direction, and you do 2 images, and you can subtract, and this subtraction is then proportional to the strength of the magnetic field induced by the currents. And so you don't measure the electric field as such, but you measure its magnetic field. But you can use it to reconstruct the current flow from it, and also potentially the conductivity distribution inside the head. Now, what are the challenges? Well, the challenges are that, because we use relatively weak currents, the resulting changes of the magnetic field of the scanner are extremely small. So we change, for example, a 3 Tesla field by less than 1 nanotesla, and that's something we have to detect. So it's a really challenging technique from an MRI sensitivity point of view. But in 2018, we managed, as one of the first, to show convincing recordings of MRCDI patterns in the human brain. And since then, we have improved our techniques quite a bit. First of all, we have actually optimized the setup to make sure that what we measure is really due to the current flow in the brain, and not because of stray fields caused by the cable. So usually what you have in electric stimulation, you have some cables which feed basically the electrodes, and these cables also create a magnetic field. And for standard cables, because of their orientation, it's actually quite strong. So this would be the simulated field which these cables create in the brain slice, which I want to measure. And this is the measurement. And you see actually that the measurement is really dominated by the stray fields created by the cables and not by the current flow in the brain. You can resolve that by our optimized electrodes and electrode cables, which have a more ideal orientation, so that basically the stray fields in the measured imaging plane is really very weak, and you see that this time you get a very different readout, which is then really due to the fields flowing in the brain and not the stray fields. The second thing is that we improved the measurements quite a bit to improve the SNR. That would be a measurement without any current flow, so it's just a noise floor, which we have. Which you see here is that compared to 2018, we have now really much lower noise floors, in particular also in regions around the ventricles, which we can now nicely image. And then we induce the current flow. Here it's from A to P, the current flow giving such a pattern of the magnetic field. Then you also see that we start seeing features here of the ventricles, which otherwise get lost. Now, with these improved techniques, what we see when we compare to simulated magnetic fields, which are then again just calculated from the simulated electric fields which we care about and what we'd like to predict, and we see that actually for a current direction in direction from A to P, when currents flow in that direction, we get always a quite nice correspondence between our simulations and our measurements. That's a really good sign. This validates our simulations. But for RL, so when we attach the electrodes here and here and lead the current flow dominantly in this direction, then the simulations differ more from the measurements. And that just really indicates that our simulated fields are less accurate for this electrode montage, where we basically have a current flow from right to left compared to an AP montage. Now, what can we do then about that? This is not so accurate. Well, actually, because we do have measurements, we can try to optimize our simulations to better fit to these measurements. And what we have developed here is a hierarchical fitting approach, where we optimize the conductivities of head models to simulate the electric fields and make them as close as possible to the measurements, but at the same time to avoid overfitting by making sure that when we go to a more complex model, then this model explains the measured pattern also statistically significantly better than simpler models. From those kinds of optimized magnetic fields, we can again look at the current flow. So that would be then a current flow which is optimized based on the measured magnetic field and compared to a current flow which we would predict from literature values. So basically when we say the conductivities of the tissues are chosen according to literature values. And you see a very clear difference in the sense that here the current flow is particularly a bit weaker than predicted here. So that's nice. It's a statistical framework which prevents overfitting. We still have one caveat, namely that because we only measure one slice at the moment in MRI, we cannot really generalize easily to other montages to make sure that the conductivities which optimize our simulations for that direction also fit for others. But that's something we're working on by improving the coverage of the measurements and also further optimizing or developing the analysis techniques. And with that, I'd like to stop and would like to thank my collaborators and co-workers, in particular also Lars Hansen, with whom I was collaborating now already for a long time on MRE's CDI, and Christopher Hansen, with whom I was now working on this recent interleaved from I.
Welcome to my talk. I'm looking forward to giving you some examples on the characterization of NIPS-induced changes at the behavioral and neural network level. My group is interested in the adaptive plasticity in neural networks for cognition, in particular in the language network. And if one is interested in systems plasticity and mapping the effects of induced plasticity, then a good approach is to combine neurostimulation with fMRI to map such changes. So with this combination, we can model and map neurostimulation effects with fMRI in healthy volunteers. This illustration shows the first approach where we use neurostimulation plasticity-inducing neurostimulation protocols applied before fMRI to map lasting stimulation after effects at the neural network level afterwards. And with this, we can address so-called rapid short-term reorganization, that is the response of the network to a focal perturbation induced by plasticity-inducing neurostimulation protocols. And this allows to answer the question how the targeted network may compensate for a controlled focal perturbation induced by neurostimulation. We can, of course, also map the effects of facilitatory neurostimulation at the neural network level. Now, a second approach is the simultaneous combination of neurostimulation and functional neuroimaging to map immediate effects, that is, the immediate neural correlates of our stimulation effects directly during fMRI, or, of course, also changes across the time course, for instance, during learning experiments. And both approaches allow to modulate task-related activity in the network of interest and also at the whole brain level, task-related connectivity, and relate this to changes at the behavioral level. So this provides a complementary insight into the induced neurostimulation effects. Since my group is mainly interested in the language network, I want to give you an example on the adaptive plasticity in the semantic network. So in this study, we were targeting a key node for semantic processing, the processing of the meaning of words and sentences with inhibitory TMS before functional neuroimaging, and afterwards healthy volunteers performed simple word judgments, semantic word judgments during fMRI. And what you can see in blue here is the effect of a perturbation of a key region for semantic processing in the left angular gyrus. So angular gyrus TMS results in a decrease of the task-related activity, not only in the stimulated left angular gyrus, but in a larger semantic network, including the anterior IFG and also the posterior middle temporal gyrus in the left hemisphere. And interestingly, we found that this remote inhibition of distributed activity in the semantic network is likely induced by an increase in the inhibitory connectivity between the targeted left angular gyrus and the anterior IFG. So using dynamic causal modeling, we found a strong increase in the inhibitory influence from the targeted angular gyrus on the anterior IFG. So it seems that TMS of left angular gyrus decreases activity in the semantic network, and this inhibition at the network level is likely mediated via an increase in the inhibitory influence between the targeted areas and other areas in the specialized network. Now, interestingly, this remote inhibition appears to be behaviorally relevant because the level of the individual inhibition predicted the individual disruption of task performance. So the stronger the individual inhibitory influence of angular gyrus on anterior IFG after TMS, the stronger the delay in the semantic response speed after TMS. So stronger individual increase in the inhibitory influence, that is more negative here, that is slower response speed after inhibitory TMS. Now, we not only observed decreases or inhibitory effects of our perturbation, but also increases in task-related activity after inhibition of the semantic network. And these increases in activity were found in these areas highlighted in red here. These are areas that are not typically associated with semantic processing, but rather with processing of the sound of words, its phonology, and also domain general areas. So such as posterior IFG and supramarginal gyrus of the phonological network and superior parietal lobe, a domain general cognitive control region. So these areas showed an increase in their task-related activity after perturbation of the semantic network. And we found that the level of the individual inhibition of the angular gyrus predicted the upregulation of these neighboring parietal regions in SMG and SPL after TMS. So the stronger the inhibition of the angular gyrus, the stronger the upregulation of these neighboring regions. And we reasoned that this likely reflects partial compensatory upregulation of the phonological network and domain general cognitive control regions in the superior parietal lobe, because the overall level of task disruption in the mean response speed or accuracy was relatively low. So this could reflect an attempt of the network to compensate for the large-scale disruption of the semantic network by recruiting neighboring regions. Now, the majority of studies that we perform with neurostimulation are significant at the group level only. This was also the case in the data that I've just shown you. So we recently reanalyzed this data using a multivariate pattern approach to map changes at the individual level. So this is the original data in each individual subject analyzed with a multivariate pattern analysis. And you can see that this difference in the activation pattern, or this shift from activity from the stimulated left angular gyrus to the neighboring supramarginal and SPL region, could be mapped in each individual subject. And interestingly, while we could show this shift of activity, there was quite some variation at the individual subject level, so individual specific response profiles. And this may indicate that at the single subject level, MVPA may be more sensitive relative to univariate approaches to map individual differences and individual specific response patterns of neurostimulation. And this may have some translational relevance because, especially in patients, we know that they often show strong differences in their individual response to therapy. So maybe in the future, one could use such multivariate, more sensitive approaches to map changes in response to therapeutic neurostimulation approaches. Now, I have recently suggested a new model for flexible redistribution in neural networks for cognition based on such neurostimulation studies, mainly in healthy volunteers. And this model includes a mechanism for compensation across different networks. So as shown in the semantic network, one idea is that if we observe large-scale disruption at a larger network level induced by neurostimulation or potentially also by brain lesions, then this may decrease the contribution at the larger network level. Now the brain may compensate via recruitment of alternative networks. This could be neighboring networks for other specialized functions. For instance, the phonological network is recruited to support semantic computations, as in the example before. And this may also include the recruitment of so-called domain general networks. Domain general functions such as attention, working memory, or cognitive control are known to support all higher cognitive processes, such as language or also social cognitive functions. And the idea is that their recruitment may to some degree help to maintain task processing. I don't suggest that domain general networks can fully replace specialized functions. Now, one question that remains to be addressed in future studies is the behavioral relevance of the interplay of language-specific and domain general networks during language processing or other higher-level cognitive processes. And this is something that we're currently working on. Now, I want to focus on the simultaneous combination of neurostimulation and functional MRI to map immediate stimulation effects and give further insight into this interplay between specialized language regions and domain general areas. Now, this is an example study that we did together with Paola Marangolo's lab from Rome. And here we were interested in the neural correlates of TDCS-induced improvements in verb learning. So we're now moving to electrical brain stimulation with the aim to improve performance in challenging tasks. So we had healthy German volunteers who had to learn low-frequent Italian verbs, a very challenging task. And this task was performed during fMRI under a nodal TDCS, so facilitatory stimulation, or sham TDCS in different sessions. And subjects received stimulation over the left inferior frontal gyrus, which is a key region for verb learning and processing. The task was such that the participants saw pictures of verb actions, and they had to overtly produced the corresponding verb, and afterwards they received written feedback. So they saw this something like to blow, had to say to blow, so fiare in Italian, and then they received written feedback. And we only had a very short training session. The actual learning took place in the scanner via repeated presentations. So this was a very challenging task. You can see the behavioral results here. This is the learning curve across both sessions, so for sham TDCS and anodal TDCS. You can see that we had repeated combinations of these pictures and the corresponding verbs. And subjects showed improvements, so a nice learning curve in both sessions, but we had significant improvements under anodal TDCS relative to sham stimulation of the task-related accuracy in the later time course of the learning procedure. Now, the main question of this experiment was, what are the neural correlates of such TDCS-induced improvement? What can we observe at the task-related activity and neural network level? So what you can see here is that TDCS induced strong decreases in task-related activity in the stimulated area and also in other language areas. So we found decreases in the left IFG, in the contralateral homologous regions, and also in other areas associated with verb processing, such as the left fusiform gyrus. It seems that TDCS induces decreased activity in specialized language areas, which likely reflects increased processing efficiency, as shown in other combined TDCS fMRI studies and also in patients with post-stroke aphasia who showed decreased task-related activity in the language network after successful therapy. Now, finally, we were interested in changes in the effective connectivity between the targeted language area and the rest of the brain. And as you can see here, using psychophysiological interaction, we found decreases between the left stimulated inferior frontal gyrus and the right anterior insula during verb learning. The right anterior insula is a key region for cognitive control, so a domain general area. And these changes were behaviorally relevant because the individual decrease in the task-related connectivity between IFG and right insula predicted the individual TDCS-induced improvements. So the stronger the decrease, the stronger the individual increase in task accuracy under anodal stimulation. So it seems that anodal TDCS decreases connectivity between a language-specific node and a domain general cognitive control region. And again, this likely reflects increased processing efficiency under facilitatory neurostimulation. So this shows that the interplay between specialized language areas and domain general areas is behaviorally relevant. Now, finally, I want to give an outlook where this could, where we could move to in the future. And one idea is to use network stimulation of densely connected hubs, which might also be interesting for therapeutic applications of neurostimulation. And one network, promising network of interest might be the default mode network. This is this task negative network that is usually active when we are not engaged in a specific task. So this is data or an illustration from a recent review summarizing previous resting state studies that could show that a single site stimulation of densely connected hub regions at rest resulted in strong effects at the network level. And a number of studies targeted different default mode regions, and in particular the left inferior parietal lobe of the default mode network. If we stimulate this with TMS, it results or induces large-scale changes in connectivity at rest across different networks, not only in the default mode network, but also its connectivity with other task-positive networks. So this strong remote stimulation effects of across networks are particularly interesting towards therapeutic applications, but their behavioral relevance remains unclear so far because the majority of previous studies focused on resting state connectivity. So what we're interested in at the moment is the effects of neurostimulation on task-related activity and connectivity, and also domain-specific effects versus domain-general effects across different networks. And on the long run, the translational relevance of such network stimulation will also be of interest. This is data from a recent study performed by Ole Numsen from our lab, and he was interested in the functional specialization in the larger IPL region. And he could derive a task-specific parcellation of the larger IPL region into different subregions, an anterior one and a posterior one in each hemisphere. And these subregions differed with respect to their predictive relevance of the task-related activity for the different subdomains. And we had the three domains: attention, semantics, and social cognition, which are all key domains of human cognition. And in particular, Ole could show that the right anterior IPL subregion had the strongest predictive relevance of task activity for the attention domain, whereas the left anterior IPL subregion had the strongest association with semantic processing. And finally, both posterior IPL subregions had the strongest predictive relevance for our social cognitive subdomain. And this shows that larger regions that are also relevant at rest can be dissociated according to their contribution to different cognitive key domains in the human brain. And this functional specialization could also be found at the level of task-related connectivity. This is an illustration of the connectivity profile for the social cognitive tasks. So both posterior IPL subregion show dense connections with the major networks that are also described at rest, for instance, dorsal attention network, other nodes of the default mode network, and the visual network. So also the specific connectivity profiles were different across the three domains. And now the next step, what we're currently working on, is to use network stimulation to dissociate domain-specific and domain-general effects when we target these different subregions. So to summarize the modeling of NIPS effects in cognition research, what does neural modeling and mapping in the combination of neurostimulation and fMRI give us? We can map immediate or lasting stimulation effects at the neural network level. This gives insight into the potential for flexible adaptation and compensation in neural networks for higher cognitive functions. We can map immediate network effects and remote effects within specialized and across different networks. And some advantage of neural modeling approaches include the increased sensitivity of fMRI measures for modulatory neurostimulation effects. We can have changes in task-related activity and connectivity and investigate their relationship with changes at the behavioral level. And the way forward, finally, may be to use network stimulation via stimulation of densely connected hub regions, such as areas in the default mode network, which may also have therapeutic relevance in the future. With this, I'm at the end of my talk. Thank you for your attention. I'm looking forward to discussing these results.